All right, let's carry on with a continuation of the nervous tissue. And this is going to be kind of the intense part of this chapter. This is where we're going to start to talk about electrical signals. And my advice to you is to take this in small parts. Don't try to just do it all in one sitting. You know, baby steps, you still arrive at the destination. So, all right, let's get started. First, let's talk about the two different types of signals that we see in neurons: action potentials and graded potentials. Action potentials are coordinated, could be for a short or long distance, but they can travel very long distances, which is important. And they have very specific types of channels that open and close in response to certain amounts of positive or negative ion concentrations. That is a voltage-gated channel. They respond to amounts of positivity or negativity, which is voltage. We also have graded potentials, and these are just for kind of short distances, and it's always diffusional. So there's no fancy channels here. It's really just kind of a gradient from the start to the stop, which is why it has to be short distances, because diffusion, it can't really get very far. So we're going to talk about those two types of signals, action potentials and graded potentials, in great detail. But before we even get to signaling, we need to talk about what triggers a neuron to even respond to any type of disturbance. And that means, how is a neuron tuned or triggered to be excitable? And that is a property that neurons have called the resting membrane potential. They have a potential at rest that basically sets them up to respond to different types of stimuli. We'll also see that there are different types of channels. Now, you know that your body can detect different types of stimuli, right? Some of those stimuli require different types of channels to actually create the sensation of a stimulus. So we're going to talk about those. But first, let's start with the resting membrane potential. This is a complicated idea because not all cells have a resting trigger point. But neurons definitely do, because they're just waiting for some kind of a disturbance. So they have to have some kind of resting state that allows them to detect something changing in their environment. And how they do that is through what we call the resting membrane potential. That means that the membrane of a neuron is actually charged. It has potential based on a difference in the ion concentrations outside versus inside the neuron. And this little device here is called a voltmeter. It's just a machine that allows us to basically see how negative or positive a space is. And we always use it to compare two different points. So in this case, if we have one side of our voltmeter on the inside of the neuron, and the other side on the outside of the membrane, what we would see is that the inside of the neuron is much more negative than the extracellular fluid. In fact, we can actually measure that at rest to be minus 70 millivolts. The key here is that it is negative. It is more negative inside the cytosol of the neuron than outside the cell. In fact, you can kind of think about the extracellular fluid as being neutral. So it's got no charge, really. But there's a lot of negativity, negatively charged ions inside the neuron. Here's how I want you to think about what produces that magical number of minus 70. First, cells are full of proteins, right? Proteins are constructed from amino acids. Amino acids in cytoplasm have a negative charge. That is just something that we have to accept. Amino acids have a negative charge. And if we had a hypothetical cell, a total hypothetical cell here with nothing else, just a membrane and a bunch of proteins, what we would measure with our voltmeter is that the negativity from those amino acids gives us about minus 150 millivolts. They are very negative. Now, this is not the right number. We know that this is not everything that produces the resting membrane potential, but it's kind of step one. So step one, cells are full of amino acids and those have a negative charge, and it's very negative. 2, we know that neurons have potassium leakage channels. Leakage channels are the kind of channels that just open and close randomly. They're very leaky. And what we'll see is that because that cell is very negative and potassium has a positive charge, the positive potassium is actually attracted to the inside of the cell. We're actually going to see potassium leak into the cell. Now, if we kind of do a running total here, if our amino acids added to the amount of potassium that's leaking in, remember, amino acids are negative, potassium is positive, this is actually going to take our overall charge difference to about minus 90 millivolts. We're actually really close to that magical minus 70 number right now with just amino acids and the fact that potassium has leaked into the cell. I will also add about potassium that leakage channels achieve equilibrium. So there is potassium coming in, there's potassium coming out. It's just kind of happy and stable, but it gives us this minus 90 millivolts. We know we're not quite there yet. We know that there is something else happening. The third characteristic of the resting membrane potential is that neurons have a little bit of sodium leakage. Now, there is a ton of sodium outside the extracellular fluid, right? Remember, in our hypothetical cell, all we have so far here is just proteins with their amino acids, and we've allowed some potassium to leak in. So there's no sodium. What happens is this concentration of sodium leaks in, it kind of forces it in, and sodium has a positive charge, just like potassium. So if we take our negative amino acids, we started at minus 150, right? We add a little bit of potassium, we got up to about minus 90. We add a little bit more sodium. This actually gives us the minus 70 millivolts. Hooray, we've done it. But it's really important that we kind of think about these conditions individually. So just to kind of recap, we've got overall negativity from the amino acids. There's some other things inside the cell that are also negatively charged, but think of them as the same kind of phenomenon. They're just producing an overall negativity. Then we have this potassium leakage channel situation where potassium is coming in to equilibrium, giving us minus 90 millivolts. There's not a lot of permeability or channels that are open for sodium. We see just a tiny bit of leakage because the concentration gradient is strong. But the good news is we have sodium-potassium pumps to return the sodium and the potassium to where we want it. This is a pump, which means we spend a lot of energy to pump sodium back out that's leaked in and keep the potassium inside the cell. Now, one of the key mistakes that I see with resting membrane potential is people assume that the sodium-potassium pumps produce the resting membrane potential, and that is not true. In fact, we can block those pumps, and it stays pretty close to minus 70. What we need those pumps to do is to maintain the minus 70, all right? It's kind of maybe a classic multiple choice mistake. What produces the resting membrane potential is the negativity from the amino acids, the potassium leakage, which kind of gets us real close, to be honest, and then a little bit of sodium leakage that does occur, and then the pumps just kind of make sure that it is maintained so not too much sodium leaks in or too much potassium leaks out. Whatever it needs, those pumps are basically online to maintain. And I don't really have anything else to add about that. All right, let's kind of review types of ion channels. There's 4. The first one we've already talked about is a leakage channel, and these just randomly open and close. So if I were to prescribe like the numbers of leakage channels, I would say that neurons have a high number of potassium leakage channels and just a few sodium ones. So that's important. That kind of helps us establish resting membrane potential, right? The next type of channel that we haven't talked much about are the voltage-gated channels. These open and close in response to a particular amount of positivity or negativity inside the cell. Now, where's that positivity and negativity coming from? It's coming from the sodium or the potassium, or maybe even a chloride channel, which is a negatively charged ion. Anything that has a charge is going to potentially change the membrane potential. Positive charge is going to make that cell more positive, and negatively charged ions are going to make it more negative. So these voltage-gated channels are sensitive to where the positivity or negativity in that neuron is. So I know you have more questions about that. Hold that thought. We've got two more types of channels to talk about. Ligand-gated channels. We have seen an example of this already in muscle contraction. The ligand-gated channel for muscle was acetylcholine, right? It was a chemical. Ligand-gated channels open and close in response to the presence of a specific chemical, such as acetylcholine, for example. We also have, this is kind of a cool one, mechanically gated channels. These open and close in response to touch. So it can physically push this channel closed, or it can physically pull this channel open, and that's how these channels are going to alter the sensitivity of a neuron. So overall, we've got these four types of channels. Leakage, that's just random, right? Things come in and out as they will down their concentration or electrical gradients. We have voltage-gated channels, which are sensitive to how much positivity or negativity is inside the cell. They'll open and close. Ligand-gated channels open or close in response to a specific chemical. And then mechanically gated channels open or close in response to touch. So here's a picture of a leakage channel. Always open, right? Just kind of flows down its gradient. If we have more on this side, it'll flow out. If we have more on the outside, it'll flow in. It's just following the laws of thermodynamics. We can't open or close these at all. We're not going to control them. Ligand-gated channels have a specific key. There's a specific chemical that basically touches that channel and then it will open it. In this case, it's opening it. It could be, it closes it, sort of depends here, but depends on what the actual ligand is. But in this case, it's acetylcholine. It's going to attach to this channel, open it, and then now that channel is open, other ions can flow through. It is not usually the ligand itself that comes through the channel. It's usually like the key that opens the door, if that makes sense. For mechanically gated channels, these open or close in response to touch. So imagine like a hair, like a follicle of a hair sitting on top of this gate here. When your hair is moved, it basically pulls this channel open and then ions flow through. And then when it's at rest, it's closed again. It's like a trap door, basically. And this is very helpful. This allows our body to see if there are any physical manipulations happening to the neuron structures outside of the neuron. So we can be sensitive to things like touch or movement or things like that. Voltage-gated channels respond to an electrical stimulus. So here's an example of a closed voltage-gated channel. We actually see this physically closed with what we call a ball and chain. This is jamming up or blocking the channel interior so nothing can get through. But what it is sensitive to is the inside environment here. At rest, it's -70, it's closed. But once it gets a little bit more positive, this ball and chain is removed, and now this channel is open. So basically, the stimulus here is the change in positivity from -70 millivolts to -50. So it's gotten more positive. Now this blocker is out of the way, and we've opened this channel for ions to move. So first thing is to just kind of consider that we have these four types of channels. We haven't even really seen where these channels are yet, but they're going to be distributed at different parts of the neuron. That's the first important thing to think about before we get talking about signals. So if you need a moment to just kind of process that, this would be a good break point. All right, let's get started with signals. We're going to start with graded potentials. Graded is a word that implies that the amplitude or the strength of this signal, this potential, can vary. It can be graded depending on how strong the stimulus is. And this is very correlated. A small stimulus results in a small potential. A big stimulus is going to result in a very strong signal. So where do these signals come from? Well, they come from ligand-gated or mechanically gated channels opening. So when those channels open, either from a chemical in the environment or maybe a physical touch to the environment, we will see those channels open, and whatever those channels allow through is going to come into the neuron. Now, I should specify that there are a lot of possibilities for what those channels might allow to flow through. It could be a positively charged ion, like sodium or potassium, or it could be a negatively charged ion, like chloride, for example. Now, remember that our resting membrane potential is minus 70 millivolts. If we start at minus 70, we are already polarized versus the outside extracellular fluid. The word polarity just refers to that minus 70 millivolt difference, okay? So if we allow negatively charged ions to come into the neuron, that's going to increase the negativity, right? It's going to make that neuron even more negative. Maybe it goes down to like minus 80 millivolts or minus 90 millivolts. It kind of depends on how much comes through. If a lot comes through, it'll get even more negative. If a little, maybe just a tiny bit more negative. Those are hyperpolarizing reactions. Hyper meaning greater and polarity referring to the difference. If it is a positively charged ion, it will come in and it will actually decrease the minus 70 millivolt difference. So if we add some positivity to minus 70 millivolts, maybe it comes up to minus 60 or minus 50. That depolarization refers to the overall charge difference across the membrane decreasing. Keep in mind that these really do determine the signal strength based on the amount of stimulus itself. So big stimulus, more graded potential, less a smaller graded potential. And these graded potentials typically occur at the top of the neuron, the cell body or the dendrites. Let's take a look at these graphically for a second. Here we're sitting at basically, let me get my little highlighter here. Here we're sitting at minus 70 millivolts. That's the resting membrane potential. And again, if you need to go back and review what creates the resting membrane potential, please do. It's important that we see the distribution of things on either side of the neuron membrane. But we have this minus 70 millivolts here, and here we actually have a small depolarizing stimulus. It's getting more positive, so something positive is coming in, a really tiny one. Here we have a stronger depolarizing stimulus. Here we have one that's kind of like plateaued for a second. Maybe it just stays around a little bit longer, and then it kind of comes back, goes away, whatever. Here's a really, really strong one. We actually get up to maybe close to minus 50. This is a really strong depolarizing stimulus. All of these things are graded potentials. Small, medium, kind of a longer one here, and this is a really strong depolarizing stimulus. Here, we'll see that we can also have a hyperpolarizing graded potential. Remember, hyperpolarizing, something negative is going to come inside the cell, and it's going to make it even more negative. So here's a small hyperpolarizing stimulus. Here's a stronger hyperpolarizing graded stimulus. And here we have a really, 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 really strong one. A ton of negativity has come inside the cell before it returns to rest. Remember, those sodium-potassium pumps are really working hard to reset that neuron as soon as it detects some kind of a stimulus. So these don't necessarily have to stick around unless the stimulus sticks around. Now, we do have some types of graded potentials that I want to mention. what we've just talked about is a receptor potential. Visualize this as happening at the dendrites or maybe even the cell body itself. These are basically looking for some kind of environmental ligand or mechanical stimulation in its environment, opening up those channels and allowing whatever that channel is for, a depolarizing or a hyperpolarizing graded potential to occur. We also have postsynaptic potentials. Whoa. Okay, postsynaptic just refers to what is on the other side of the synapse. So remember, the synapse is where an axon terminal is going to interface with a second cell. So actually, this is a muscle contraction right here. We have a postsynaptic potential. The acetylcholine is the ligand. It gets diffused across the synapse. It binds to a ligand-gated channel, and then it opens up sodium channels and then excites the muscle contraction, right? That's an example. Now, postsynaptic potentials in general occur when a neurotransmitter binds to a ligand-gated channel. However, those ligand-gated channels, just like we saw before, can open up a positive ion channel or a negative ion channel. If it is going to stimulate a depolarization, and by that I mean a positive channel has opened, we would call that an excitatory postsynaptic potential. We usually just refer to those as EPSPs, excitatory postsynaptic potentials. If it opens up a negative ion channel, we would call that an inhibitory postsynaptic potential, or an IPSP. And it is very important at this point that you see that neurons can either be made positive or made negative. And we will see that this allows nervous tissue to tune into information, right? We can stimulate a neuron to create an impulse and then travel that impulse down the axon and release neurotransmitter. We can also turn neurons off through hyperpolarization. And there are times when we actually want to quiet a neuron down. Like maybe we want to paralyze a muscle from contracting momentarily, or we want to have both types of control. So postsynaptic potentials can happen in either directions. They can be excitatory and create a depolarization event, a positivity event, or they can be inhibitory, where they would open up negative charged channels and basically make that neuron harder to stimulate. Now, let's talk about the action potential. The action potential is going to start when we have a graded potential at the receptor of the neuron depolarize. That's the first thing you have to appreciate, is that graded potentials have to occur first. So we're going to get diffusion of a depolarizing or excitatory stimulus. Action potentials are always excitatory, right? If we inhibit with a graded potential the top of a neuron and we hyperpolarize it, we're just basically taking that neuron offline. But with an action potential, we actually want to transmit information that is important. So we're going to excite that neuron with a depolarization of that top end of the neuron. So the dendrites and the cell body are going to depolarize, and it needs to depolarize to threshold. What is the threshold? The threshold is a magical number where our voltage-gated channels open. Where are those voltage-gated channels? They're all the way down at the axon hillock. So we actually have to get a graded potential that is strong enough to trickle from the dendrites through the cell body to the top of the axon hillock. This requires up to minus 55 millivolts of positive stimulus. So we basically need about 15 millivolts of excitatory, positively charged ions to diffuse through the top of the neuron to get to the axon hillock. That is the threshold. The threshold now is going to activate the voltage-gated sodium channels. Those sodium channels open. And I want you to just kind of visualize where we are here. We are at the axon hillock. we've opened up these sodium channels, and there's tons of sodium outside the cell. Tons. It's just waiting to rush in. So when that sodium rushes in, it is rapid, and what we see is this like skyrocket of positivity. It goes from like minus 55 millivolts to plus 30 millivolts. This is a huge influx of positive charge. What happens at that peak of positive 30 millivolts? What we'll see is two things, and these are both important. One is those sodium channels inactivate. Remember that little ball and chain? That little ball gets stuck inside the channel, and that channel is actually offline for a second. Well, I should say millisecond. The second thing that's kind of happening is the potassium channels. There's also voltage-gated potassium channels that we haven't really seen yet. This now is an environment where the cell is full of sodium and it's still full of potassium from resting membrane potential, right? So the potassium really wants to exit. It really wants to push itself out. And as the potassium leaves the cell, it actually repolarizes because the positivity is leaving the neuron. So it's becoming repolarized. And these channels are just kind of little slower than the sodium channels. So this always happens after the voltage-gated sodium channels are activated, and they kind of miss the cutoff here. They'll actually allow more potassium to leave than the resting membrane potential. So we really do need to return back to resting membrane potential by employing those sodium-potassium pumps to kind of pump everything out into the right place, because the sodium and the potassium are now reversed. We have a cell full of sodium, and then we've pumped all our potassium out, so we want to reverse that and pump that stuff all in. Perhaps a graph will help you visualize this. First, here we are at rest at minus 70 millivolts. Here's our stimulus. This is our graded potential. It needs to be an excitatory or depolarizing stimulus, right? Because if it was hyperpolarizing, it would just make those voltage-gated channels harder to open because it would be more negative. They always open at minus 55 millivolts. So we need a depolarizing positive graded potential to reach minus 55 millivolts at the axon hillock. This is the only place where we see the voltage-gated channels, but the first ones to open are the voltage-gated sodium channels. And it's like, whee! All this sodium is rushing into the cell. Right here at plus 32, things happen. Those voltage-gated sodium channels close, and potassium channels open. Potassium wants to leave the cell, and as it leaves, it is getting more negative because potassium carries a positive charge. Little extra hyperpolarizing burst here because those channels just don't know when to quit. And then we have our sodium-potassium pumps basically restore resting membrane potential. That's an action potential. So it's like this little, it's almost like a heartbeat. It's like this little sodium, potassium, sodium, potassium, sodium, potassium, and then it resets between each firing. The refractory period is very interesting, I think, but it serves a couple purposes. When we talk about the refractory period for neurons, what we're actually talking about is the time that it takes to basically get a second signal possible. And we know that those sodium channels are physically inactivated. So when those are blocked, we're in an absolute refractory period. They're not going to work again. They're just sort of turned off temporarily. We have to kind of reset the neuron a little bit and get that ball and chain unstuck from the channel pore for those to reset and actually be re-stimulatable. That's the relative refractory period, and that just refers to, yeah, we might not be back perfectly to resting membrane potential, but if we have a really strong second stimulus, it might exceed that second modified threshold and get a second firing. But refractory period is really just referring to how much time does it take to reset, and there's kind of two parts to the absolute and the relative refractory period that I want you to know. One is like channels are blocked, can't go, and the next one is channels are not blocked, but we're still not back to where we want to be. We might limp along, but it's going to need to be really important for us to fire again. That would be the relative refractory period. Now, the thing about conduction of the action potential is there's kind of these two different ways that it can go. And we'll see this in the structure of the neuron. Here on the bottom, we have an unmyelinated axon, which means there's no Schwann cells, there's no oligodendrocytes, there's no myelin, there's no insulation at all. This is basically just a domino effect. As one area of the axon undergoes an action potential and like the sodium comes in and it becomes more positive, it's going to basically stimulate its neighbor to do the same thing. And we just see this rolling wave of neighboring voltage-gated channels, one after another, ripple that signal down the length of the axon. Now, because this is not insulated, we can sometimes see this fail. We will sometimes see, oh, this channel didn't work very well, and then, oh no, there's not enough to depolarizing the next sequential channel set. That would mean that the signal would be deteriorated. This is why unmyelinated axons have to be for short distances only. It requires a lot of energy. It's really slow. The channels have to be like really close together, so there's a lot of channels, but it's basically like a domino effect, if you can visualize that. The better alternative is to have insulation. This works just like an insulator would. We're going to start at the axon hillock here, right? So we've basically received our graded potential stimulus down the dendrite, through the cell body, down to the axon hillock. And this is where our first voltage-gated channels are. If we get up to a positive stimulus up to minus 55 millivolts, we're going to start to see our first action potential occur here. So we're going to see sodium come in, potassium go out, and then what happens is where there is insulation of the myelin, that wave of positivity from the sodium just shoots through that channel. And then it arrives at a node of Ranvier, which is a naked part of the axon. At that node, there are more voltage-gated channels. So the positive wave goes through the tunnel, and then it recreates the action potential at the node, and then it goes through the next tunnel, and then it has the next population of voltage-gated channels. It reproduces an action potential through the channel, et cetera, et cetera, et cetera. It's like a chain reaction, right? But the key here is that the action potential is regenerated at each node. So the action potential is fresh. It's just as strong as it started. This is what we call saltatory conduction. Saltatory means that it skips between the nodes, regenerates the action potential, and by the time it reaches the end, it is just as strong as it was in the beginning, which is a huge advantage if we have to transmit information over long distances. But clearly, we will see examples of unmyelinated short little axons and myelinated ones traveling differently. Instead of continuous conduction, it is saltatory, or it skips between the nodes. Now, what happens at the end, right? The first possibility here is an electrical synapse, which is a gap junction. So remember, a gap junction is a tube of cytoplasm. We saw this in cardiac muscle, for example. There are times when we want a signal to go from one cell's cytoplasm directly into the next cell. We don't even want to mess with a regeneration. We don't want a neuron in the way. We want it to just happen really, really instantly. That is the case of an electrical synapse or a gap junction. It's a tunnel of cytoplasm. It just rips right through it. This will synchronize any type of effector so that it's all getting the signal at exactly the same time, which is super advantageous, like in the heart, for example. We could also have what's called a chemical synapse. So in this case, we have our neuron, we have our excitatory graded potential here, sitting at minus 70. Some positivity is trickling in. If we get up to our threshold at minus 55 millivolts, we're going to generate our action potential. It'll be like shooting through those myelins to the nodes, regenerating, it comes to the end. And what happens here is that there's actually a different process at the axon terminals. We'll have this wave of positivity coming from the action potential. It's coming from the axon, right? That positivity arrives at the axon terminal, the synaptic end bulb, and what it does is it opens voltage-gated calcium channels. So we haven't talked about calcium yet, but calcium is actually quite special. Calcium comes in and it stimulates these vesicles of neurotransmitter to exocytose and then travel across the synapse. So a slightly different process happening here at the axon terminal. We have voltage-gated calcium channels. Those open. The calcium is basically what propels these vesicles to exocytose and release the neurotransmitter into the cleft. This is just like the word version of that. Nerve impulse arrives at the synaptic end bulb. We'll see that wave of positivity or the depolarization opens the voltage-gated calcium channels. Calcium comes in. It causes the neurotransmitter vesicles to basically merge with the cell membrane and release. That neurotransmitter diffuses across the cleft, binds to the receptors on the postsynaptic cell, and then we know that there's two options. It can either be excitatory or inhibitory on the postsynaptic cell. So it can either depolarize the second cell or hyperpolarize. Whoops. Sorry, just knocked something over. I like to do this stuff standing up, and I'm just like, I'm a mess today. But anyway, I can't stop now. This is too good, right? The neurotransmitter causes those channels to open, and that will either depolarize or hyperpolarize the second cell. If it is depolarizing and opens a positive channel, we'll see a new action potential. And then it just kind of carries that forward. Okay, just a couple more complications to talk about. Now, your brain is definitely tuned for action potentials. But your brain is also going to want more information than, oh, I have an action potential occurring at this location. It wants to know if it's like super important or, hey, do I need to like pay attention to that? Maybe I can ignore it for a while. We will see that there's other ways of transmitting information, even though action potentials are always the same, right? Action potentials, once it starts, same action potential at the end. An action potential is just like every other type of action potential. So how does the brain know? Well, there's kind of two options. One, we could have one neuron continue to just keep firing. It's just like action potential, action potential, action potential, action potential. So with time, it repeats. That's temporal or time summation. And basically that alerts your body that it wasn't a misfire. Something is really happening there. Pay attention. Like someone is giving you an injection. Ow, 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 ow. Right? That's temporal summation. But it's very localized. It's very small. One neuron even. Spatial summation is where maybe multiple neurons in a general area are all firing at the same time. Like, oh, I've got neurons here, 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 here. Well, that's not a misfire. Multiple neurons means that something is really going on. Perhaps if one neuron fires once in a while, or maybe one neuron from an area fires, the brain will ignore it. But if one neuron fires over and over, or if multiple neurons fire at the same time from the same general localized place, then the brain is going to know about it. So here's an example of what I'm talking about in terms of summation. When we have temporal summation, this little neuron right here in the middle is like beep, 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 beep. And then the second cell is like, okay, I've reached threshold now that you fired 10 times. I'm going to send this information to the brain. Down here, we've got these three neurons. They're all firing. And now this second neuron is like, okay. I needed all three of you guys to tell me that this was real, but I'll go ahead and relay that information. Those are two really important ways that the brain can filter out unimportant or inadvertent information. We can either, pay attention to one neuron that's just going crazy, or, hey, we've confirmed multiple sensors are all firing at the same time. We'll go ahead and let the brain know. But it is important that we filter out some of that nonsense because, of course, action potentials can misfire. Of course, sensory neurons can, sometimes receive small information that, your brain doesn't really need to think about until it gets really major. I do want to mention removal of neurotransmitter because once we've dumped neurotransmitter into the synapse, we need to do something about it. And there's kind of three ways we can deal with it. We can allow it to just diffuse into the area, which is the case of adrenaline, right? This is why adrenaline stays in your system for hours. You're just kind of still, a little bit stimulated because it takes a while to diffuse. We can have enzymes degrade that neurotransmitter. We saw that with acetylcholinesterase. That's very fast. It's very helpful to have an enzyme just come in and be like, oh, I got you, clean up right away, done, reset, perfect environment. It also is really important if we don't want to overstimulate the second cell, if we want to really stop it from receiving a signal instantly, we need some kind of enzyme to degrade the neurotransmitter. Or we can actually pump up stuff by the same neuron that released the neurotransmitter. We can suck it back up like a vacuum cleaner. And this is basically selective reuptake. And I find the word reuptake a little bit confusing because it's almost like redundant. But it is bringing it back inside the cell. And this is actually how antidepressants like Prozac and different types of antidepressant therapies work, is they're selective serotonin reuptake inhibitors. So antidepressants basically inhibit the uptake of serotonin. Serotonin is the neurotransmitter that modulates mood. So you can stay happy longer if you have more serotonin in the synapse. It just kind of lets it linger a little bit longer. If you've heard that terminology, it's actually activating the uptake mechanism. Finally, I just wanted to mention that we have different types of neurotransmitters. Acetylcholine is so important. It is in a category all by itself. Acetylcholine we see at the neuromuscular junction, or the NMJ. So this might have different types of receptors for acetylcholine, and we'll talk about that later in this class, but know that the receptors can maybe change how they respond, but acetylcholine as a molecule is its own type of neurotransmitter. Amino acids can often be neurotransmitters. And one of the interesting things about amino acids, and this is kind of a generalization, but it's okay for now, amino acids have a negative charge, right? Amino acids are often hyperpolarizing types of neurotransmitters. So if you wanted to have an IPSP, or an inhibitory postsynaptic potential, releasing an amino acid as a neurotransmitter is the way to go. There's also biogenic amines. I remember amines as having amine at the end of their word. So like dopamine, catecholamines, which include like epinephrine, norepinephrine, that kind of stuff. Those are in different classifications. Oh, histamine, that's a really interesting one. Histamine moderates like allergic reactions, like inflammation responses, basically. Those come from these neurotransmitters, the biogenic amines. We also have neuropeptides, which I find very fascinating. Substance P is P for pain. I find this really interesting that your cells have within them all of these different substances, some of which are neuropeptides. And if the cell is destroyed, right, if all the cytoplasm leaks, you would want to know that, right? So when your cell is destroyed, some of the normal interior components, such as substance P, that has no role when the cell is intact, leak out and are neurotransmitters to transmit pain. And this is actually how you detect pain, is like your cells are actually exploding and those interior molecules leak out, and it's a neurotransmitter that signals pain. Endorphins are in this group. There's also gut-brain peptides. And I think these two are really interesting because they tend to have a sense of euphoria associated with them, which means they make you feel good. Like a good workout, a good meal. Like it makes you happy. Those are feedback loops that you definitely get from neuropeptides. Opposite of pain, perhaps, but definitely the same kind of chemical structures. ATP can be a neurotransmitter, and there are some gases, carbon monoxide, which I do not recommend, as it is going to interfere with your ability to bind oxygen to your hemoglobin, but it is like a poisonous gas. Nitric oxide also. Nitric oxide kind of is a vasodilator. It can also make you feel relaxed. So like laughing gas, when you go to your dental hygienist and you're feeling anxious or something, they can administer different types of neurotransmitters to calm you down. But I think it's interesting that they're not all chemicals. Some of them are actually gaseous molecules and lipids. So there are all sorts of different types of chemicals that can produce neurotransmitters. But the key is to kind of understand, well, what do those neurotransmitters do? And we'll talk about each one of them as they come up.